Hello guys, my name is Mercy Japosgay from KSTVET. I'm going to take you through the following. Our today's lesson is introduction. The introduction part is understanding mutation in biology. So, welcome to today's lesson on mutation. Mutations play a crucial role in the diversity and evolution of living organisms. In this lesson, we will explore what mutations are, the different types of mutation, their causes, and the consequences they can have on an organism. So, what is mutation? Mutation is a change in the DNA or RNA of a living thing or a virus. DNA and RNA are molecules that carry the instructions for making and controlling living things. These molecules can change by mistake or by exposure to radiation or chemicals. These changes can be passed to the next generation of cells or organisms. Mutation can have different effects depending on where and how it happens. How do mutations occur? In this case, there are two main ways in which mutation can occur: germline mutation and somatic mutation. Under germline mutation, it happens in the cells that make eggs or sperm. This means that the mutation can be inherited by offsprings. Germline mutation can cause genetic diseases such as cystic fibrosis or sickle cell anemia. The next one is somatic mutation. This happens in any other cells of the body. This means that the mutation is not inherited by the offspring, but it can affect the function of the cell or tissue where it occurs. Somatic mutation can cause cancer or other disorders. Mutation also differs in the way that the genetic materials is changed. Mutation may change in the structure of a chromosome, which is chromosomal mutation, or just change a single nucleotide, the gene mutation. Let's delve into the details. First, there is chromosomal mutation. A chromosomal mutation is a change in the structure or numbers of chromosomes in a cell. This mutation can affect the function and expression of genes and cause various diseases and disorders. There are different types of chromosomal mutation, such as deletion, duplication, inversion, translocation, aneuploidy, and polyploidy. I will explain each. In deletion, a part of chromosome is removed or it is lost. In duplication, a part of chromosome is copied and inserted into the same or another chromosome. In inversion, a part of a chromosome is reversed in orientation. And in translocation, a part of a chromosome is exchanged with another chromosome. In aneuploidy, an abnormal number of chromosomes in a cell, either more or less than the normal amount. And the last one is polyploidy, whereby an abnormal number of chromosome sets in a cell, usually more than 2 sets. The next subheading is chromosomal mutation can be caused by various factors. The causes of chromosomal mutation, such as errors. The first one is errors in the cell division, such as non-disjunction or unequal crossing over. The next one is exposure to radiation, chemicals, viruses, or other environmental agents that damage DNA. The last is inheritance of abnormal chromosomes from parents or ancestors. In continuation, chromosomal mutation may cause several disorders in humans, and this includes Down syndrome, Turner syndrome, Klinefelter syndrome. The first one, Down syndrome, is where an individual has 47 chromosomes due to non-disjunction of chromosome 21. It is also known as trisomy 21. Individuals with this condition have slanted eyes with flat and rounded face, mental retardation, large tongue, and weak muscles. The Turner syndrome, it brings about a sterile and abnormally short female. It occurs due to the loss of one sex chromosome, i.e., the individual has one X chromosome instead of two. With the case of Klinefelter syndrome, it results in a sterile male who may be mentally retarded. It happens due to an additional X chromosomes, i.e., the individual 47 chromosomes. Let's have a look at gene mutation. Gene mutation is also known as a point mutation, is a genetic mutation where a single nucleotide base is changed, inserted, or deleted from a DNA or RNA sequence of an organism genome. Point mutations have a variety of effects on the downstream protein products, consequences that are moderately predictable depending on the specifics of the mutation. There are different types of point mutation, i.e., transition, transversion, synonymous, non-synonymous, missense, nonsense, and frame shift mutations. Each type has a different impact on the structure and function of the encoded protein. Point mutation can be caused by various factors such as errors during DNA replication, exposure to mutagens such as radiation, chemicals, or viruses, or spontaneous decay of DNA bands. So, at some points, mutations are harmless or even beneficial, while others can lead to diseases or disorders. For example, sickle cell anemia, which is caused by a point mutation in the hemoglobin gene that changes a glutamic acid to a valine in the hemoglobin protein. This causes the red blood cells to become sickle-shaped and prone to clogging blood vessels. We have cystic fibrosis, which is caused by a point mutation in the CFTR gene that deletes a phenylalanine in the CFTR protein. This causes the protein to be degraded and unable to transport chloride ions across cell membranes, leading to thick mucus accumulation in various organs. Thank you so much. That was my biology presentation. Have fun while watching it.